So I think if we have seen now the landscape of possible nanomedicine applications like presented by Laurent, you have seen that fields like drug delivery and even now, nanobiotics is working on nanoparticles that can as itself used as a drug. You have to look and discuss different paths of development. If you go for drug delivery, the active compound is something else, is a small compound or maybe an antibody as we know of. And then what you called nanotoxicity was the term you used, right? Interesting. The translation of nano from Greek is small, so it's small toxicity. So interesting interpretation, maybe. So then I think the normal drug development rules and processes apply. You need to show in the normal systems, models, whatever, that your compound with this delivery system is not giving any additional harm to animals, systems, and finally the human. I would foresee if you are using a very new technology, maybe new materials, that the authorities will maybe request special procedures. If you think about the phase 1 development, then those escalation studies would for sure maybe start with lower amounts and lower concentrations of that system that you have developed compared to other drug delivery systems that have been proven to be in some way safe, and let's say only a new drug is then to be tested in humans. So I hope this gives you a bit on this side a flavor what might come and what might in addition be applied by authorities or by other regulatory bodies to your development. If your compound itself is the active part, I guess in some way the same rules apply. I guess maybe the authorities will ask for additional studies to understand more if this particular nanoparticle, nano whatever, is safe and can be applied. But I think this is, as for any true innovation, a kind of challenge that you have to face in your development path, and that is not and will not be different for a nanomedicine approach. I hope that covers some of your aspects, but please add.
Even if you are using nanoparticles which we know are safe, as for instance liposome or albumin, you will change the PK characteristic of the active ingredient. So in this respect, you have the potential nanotoxicity per se, but you will also modify the toxicity or the safety profile, and we hope most of the time we will improve the safety profile. But you can also have some new toxicities which were not existing with the NAIC molecule, which will appear because of the combination within nanoparticles. So you have this nanotoxicity which can be directly related with the nature of the nanoparticle itself, but also the consequence of the modification in the product characteristic you will be induced.
Maybe just one comment. I have said in the beginning that at the end it's only a nano in medicine. And when you talk about toxicity and safety, that's true. That's even more true than this. Because when you look at a new product that will be applied in human, you're not looking at the product itself. You're looking at the patient. And what you want to see if the product is harming the patient, not what's happening to the product itself. The patient is the key parameter, is the key point of developing a drug. So at the end, the endpoint you're looking at to look at safety in animal or in patient should be the same. You want to guarantee the safety of the patient. Being said, having nano object may require some different experiment, additional or different. Not much additional, but sometimes a bit different experiment to prove or to measure the endpoint. But the endpoints itself, it's the same. That's what is key. And to answer completely your question about the price, At the end, all the studies we have made up to the clinic, they are the exact same price that for any drug. The only difference, and especially for manufacturing, has been about developing new qualification method for the characterization of the product itself. But beside this point, all the experiments have been really quite similar to any drugs.